Next, I will talk about the sequence quality assessment or quality control. So when you get your data from sequencing core, the data is usually in FASTQ format. For one read, it will contain 4 lines. The first line is the sequence ID. Second line is the sequence, ACGT. And the third line, it used to be same as the first line, and now it's just simplified to be a plus sign, and it's quality ID. And the last line is a quality score. So if you have 100 million reads, then you will have 400 million lines in your file. And if you have pair and read, you will have 2 files. The first file is ending with 1, is the read 1, and the second file name is ending with 2, is a read 2. And each read in each file is in their corresponding position. And what is a quality score? Quality score is also called the Phred score. It's calculated by this equation. P equal to 10 to the power of negative Q divided by 10. So the P is the probability that a base is called wrong, is being called incorrectly. And if the probability is 1 in 1,000, that means if in 1,000 base there is 1 base that is incorrect, then the Q score is the Phred score is equal to 30, because 10 to the power of negative 3 is equal to 1 over 1,000. And in the file, this is actually not Q, it's the number, but instead is converted to ASCII2 code, and it's by this equation. So for example, if we get a Q equal to 30, then the ASCII2 code is 30 plus 33 is 63. And if you look at the table, or if you convert it in R, just the print by this command, it will give you the ASCII2 code corresponding to 63 is 3, and the ASCII2 code corresponding to 107 is G. So generally speaking, if you have a file and with all the capital letters, then you will know that, okay, my quality read quality is pretty good, in a very good range. And if you look at the fourth line, it's all like Arabic numbers, then the quality is, you need to worry about your sequence quality. And if you want to know what, you know the letter, you read the letters, and you want to know what is the Phred score Q, or you can just use this function in R to convert this letter or the ASCII2 in the ASCII2 code into the corresponding number, and you minus 33, then it is your your Phred score. For example, in this case, your Phred score is 97 minus 33 is 64. So your file contains like if you have 100 million reads, then your FASTQ file is gonna be 400 million lines. Then how do you do quality control for each read? There is a software called FastQC. It's very popular. It's very generally used to do the quality control for FastQ file. So after you run the FastQC, you will get this output. First thing you need to look at is the Phred score Q over the position in each read. Remember when we do the sequence, we start with the first one, and then usually it's getting the sequence score is getting worse and worse when you read is getting longer and longer. So this is an example of good quality, and in the good quality one, most of your read position has a Phred score beyond 30, and just in the very end of the read, it has some outlier dropped into the yellow range, but this is acceptable because the median is still in the green range. And if you have the majority of your read or the rule of thumb is if it's 20% of the position in the read has the quality, a Fred quality score less than 20, then you need to get rid of this read. So that is the per base sequence quality. The second plot you need to look at is the per sequence quality distribution. So ideally, most of the read, this is a histogram of your read. So ideally, most of the read should be beyond 30. Here is the mean sequence quality. It should be beyond 30. This is a good example. But if you have some read that has low score, if you have another peak at lower than 20, then you know that okay, I have a chunk of read in my sample, and I need to get rid of those bad quality reads. And the third thing you need to look at is the nucleotide content per position. In this plot, the x-axis is the position in a read from 1 to 40. The y-axis is the percentage of the nucleotide content, is ACGT percentage averaged over each position. In a random library, we should expect there is no or little difference between the bases of a sequence run. So the line is in this plot should run parallel with each other, and the relative amount of each base ACGT should reflect the overall amount of this base in our genome, usually around 25 each. And in any case, they should not be hugely imbalanced from each other. It issues a warning if the difference between A and T or G and C is greater than 10% in any position, and if it's greater than 20%, this will result in a failure. So like this is a bad quality one, it fluctuates a lot, and there are some big variations between T and A here in those positions. So, but it's worth to mention that in RNA-seq library preparation, because there is they use some priming by random hexamers. So for RNA-seq library, there will be always some biased sequence position at the start of the read, and we can just ignore this warning or failure for RNA-seq data analysis. And another thing you can look at is the per sequence GC content. GC content should have a pretty normal distribution, and it's peaked at the position that same as the GC content in your genome. So if it's a human genome, it should be fairly around 50% or 48%. And if you have a bad quality sample, the expectation is the theoretical distribution is the expectation, is the blue line, and the red line is in your sample, the real GC content. If it has a sharp peak, usually it means it's possible is adapter dimers contamination, and if it's too broad, then it is maybe a contamination by another species.